Bismillahirrahmanirrahim. Assalamu alaikum. Dear student, today we are going to start a new topic that is related to the blood types, blood grouping, and how the transfusion of blood take place and other topics. Today we will discuss the blood grouping in detail. Regarding multiplicity of antigen in RBC, the surface of blood cells are covered with glycoproteins which project out into the bloodstream. These glycoprotein possesses antigenic properties and are called agglutinins, agglutinogens. These can be detected by antibodies which are named as agglutinins. So dear students, for the blood grouping, we have certain antigens in our body as well as certain antibodies by birth. These specific antigens, they are present on the surface of the RBC while the relevant or concerned antibodies, these are circulating in our body. Regarding the blood grouping, these antigens named as agglutinogens while the antibodies circulating in the blood that are called agglutinins. Whenever we talk about the antigen and antibody, we have antigen antibody reaction. So in our body we have certain antigens and also certain antibodies. They may react, but this not happens normally. What thing is going to protect our body from this antigen antibody reaction? How our body is protected? For that purpose, we have a certain law which is called the Landsteiner's law, which is named on the scientist Landsteiner who discovered these ABO blood group system in 1901. So, according to that law, if an agglutinogen is present in the red cells of the blood, the corresponding antigen must be absent from the plasma. So, if there is any specific antigen is present on the surface of the RBC, its relevant antibody will be absent from the circulation, from the plasma. So if the agglutinogen is absent, the corresponding glutenin, that is antibodies, must be present. So we have antigen and antibody in our body, but they are not corresponding with each other. These antigens and antibodies, they are of different types, and because of that reason they cannot react with each other. On the 120th birthday of this great scientist, Google celebrates his birthday and post this specific post on their page as the celebration of his birthday. So, what is next? That is how we group different types of the blood. So, for that purpose, we have certain blood group systems. Commonly, 30 occurring antigens and hundreds of other rare antigens have been found on the surfaces of the cell membrane of the RBC. Two particular types of antigens are more likely to cause blood transfusion reactions. Blood transfusion reaction means mismatched blood. What happens if the antigen is, if we don't care about the grouping of the blood, whenever we donate blood to someone, there may be chances for mismatch. So, about the OAB system, the most important ones used for the blood typing during blood transfusion. There are also rare groups like MNS system, Calculus and other systems. But according to the OAB blood type, in that we have A and B, two types of antigens. Two antigens named as type A and type B may be present on the surface of RBC in large proportion. These agglutinogens are inherited, so people may have neither of them on their cells. They have one or they may have both simultaneously. And this is the basis for grouping of blood in the OAB blood system. So if considering two antigens type A and type B, we can have four types of the blood groups. Group A, O, B and AB. In our group A, we have antigen A and you must note it that according to the antigen we are going to label it, the group, the name of the group. So in the group A, we have antigen A, which is present on the surface of the RBC. And as we have A antigen, so here we will have anti-A, that is B antibody circulating in our circulation or in plasma. Antigen is always present on the surface and antibody is circulating in plasma or in circulation. For the group O, we have no antigen. So if there is no antigen, neither A, neither B, we will label it as group O. And as we have no antigen here, so in circulation, both type of antibodies, A and B, both they will be present. In group B, we have the antigen B, and in group AB, we have both type of AB antigens. The same with much more explanation and also showing the RH group, but I will discuss RH group later on. So at this stage exclude RH positive or RH negative, just concentrate the ABO system. The blood of donor and recipient is classified into four major blood types. So we can have the group A, group B, group AB or group O. For the blood type A, we can have antigen A on the surface of RBC and anti-A antibodies, that is B. This one, the B antibodies. So they are arranged in this pattern and deciding the blood group of anyone. Again, you can see the different types of the blood group. Now, what is the genetic basis for this type of the blood system? In this ABO system, we have the presence or absence of these antigens According to that they are located on the long arm of the chromosome 9. The gene has three variants or alleles: allele A, allele B, or O allele. So, the allele A encodes for an enzyme called N-acetyl galactosamine transferase, which makes A antigen that confers blood type A. So, when this encodes, the enzyme A antigen is formed and going to be attached with the surface of RBC. For the B group, we have B allele encodes for another enzyme, D-galactose transferase, that makes the B antigen. And O allele lacks both these enzymatic activities. This is the thing you can see how the formation of A antigen takes place, and it is for B antigen. So in this way, the formation of A or B antigen, they are formed, they are attached to RBC. Now if they both are attached or they both may be absent, one of them is attached, either one of them, either A or either B, either both are absent, either both they are present. So in this way, we can have different types of the ABO system. There are two copies of these genes. If both are A allele, then genotype will be AA or A and A group, and same for the others. It's better that I should show you the next this one table, in which a very nice combination of these alleles, description of complete description of these alleles is there, either AA or AO. SO is nothing. So if AA, the final blood group is A, or A and nothing, then in this. So in this way we can have these combination. So if the blood group is A, the alleles may be either both AA or A and O. And same for the B group. For O, we can have combination of OO, and for AB, both these they are simultaneously present. Because each person has only two sets of chromosome, only one of these allele is present on each of the two chromosomes in an individual. So the presence of three different alleles means that there are six possible combinations of these alleles. Same with I am showing that what will be the antigens and what will be the antibodies. This is the summary for blood grouping, in which I am showing all the types of the antigens and antibodies, what are their shape and how they are arranged. Antigens present on the surface of RBC and antibodies present in plasma and circulating in our body. Individuals with type A blood group can receive blood from donors type A and type O, and this is because I have shown you that specific table that A group have both AA or AO alleles, genotype and phenotype. So we can have A and O for blood type A, and individual with type B, we can have B and we can have type O blood which can be received. So by this we can conclude that we can have the universal recipient and universal donor. Recipient, that is AB blood group, because it can receive any type of the blood. Individuals with type AB blood can receive blood from donors type A, type B, type AB or O blood because they do not contain agglutinins. And this is concluded through the previous table which I already shown you. Individuals type O blood group can receive blood from donors of only O. Individuals of type A, B, AB and O blood can receive blood from donors of type O blood, and it is called the universal donor as it lacks any antigen. This is a bit comparison between the percentage of these group worldwide and Pakistan. You can see worldwide this is the ratio, while in Pakistan we have this ratio. In Pakistan we have the highest blood group B, as compared to worldwide we have O, and it is very nice and God gifted because it is universal donor. Regarding agglutinins, type A blood contains type A agglutinogens and anti-B agglutinin, and same for other groups. I already discussed all of them. Lastly, we have the ratio of the agglutinin at different ages. 2 to 8 months after birth, an infant begins to produce agglutinins. Maximum titer is usually reached at 8 to 10 years of age, which gradually declines throughout the remaining life. Through this graph, I am showing you that anti-A agglutinin in the group B and O and anti-B agglutinin in group A and O, and age of the person here, and average titer of agglutinins here. So this is anti-A agglutinin and this is anti-B agglutinin in group A and O. With this, I complete the discussion about blood grouping. Inshallah, next we will discuss the blood transfusion as well as the RH group. So for that time, thank you and Allah Hafiz.